Okay. Did anyone have any questions about yesterday's lesson? I don't know if you got. I didn't check to see how many people actually looked at it yesterday in YouTube, but it always comes up in YouTube. It usually takes an hour or two for all the processing and downloading to get it up there. If I forget, obviously, please do remind me when you have a chance. So I want to just jump. I want to jump right in. I mean, yesterday we proved that what was the big conclusion from Beetle and Tatum? What was the big conclusion of Beetle and Tatum? You can type it. Good. So one gene, one enzyme. And as you can imagine, there's a lot of different ways. Sorry, my cat. Say
So cute.
So there's a lot of different ways it could have played out, right? It could have. It didn't have to be that. In terms of that actually what happens is that a gene that genes code for enzymes. And the genes code for enzymes. Okay, so now we're going to dig into transcription. And what I'm going to do is we're going to play a video twice. And you're going to fill out the next page. So we talked about the different types of RNA, and now we're going to talk about transcription. And as we talk about transcription, is the making of RNA from DNA. And we're going to start to go into details in terms of which enzymes are involved and what the DNA actually looks like. You have this video as well. I'll play it and stop and start it, and you can fill out the questions below. I did realize I made a typo in question 1. I said, what is the transcription of a gene or genes called? I said, what is the area where a transcription of a gene or gene is called? What is that region called where transcription is called? So I apologize for that. Here we go. And
yeah. Dr. Hornstein, can you repeat that one more time? Dr. Hornstein, can you repeat that one more time?
Sure. Number 1 is about the region where this, what region on the DNA is this called where transcription is occurring?
Thank you.
Okay, here we go. And we're looking at bacterial DNA here. And as we'll see, eukaryotic DNA is more complex, but it's bacteria because it's the simplest and it gives the best idea of what's going on. And then you can sort of build on it, becomes more complicated. But we want to start with this. And please mute yourself. I'll wait. Okay, thanks. Here we go.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA.
So we call the operon the entire area of the DNA in a bacterium. We're talking specifically about bacterium, where genes are transcribed. It's starting with this yellow region, which we're going to learn is the promoter, and this is the terminator region, okay? And so together, this whole thing makes up the operon.
Leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site.
So now, this yellow site here is called the promoter sequence. And we call it the promoter sequence because it promotes the transcription of RNA, right? And the sequence always has very specific nucleotides there because this is the region where the enzymes are going to initially bind. However, transcription this is where the enzymes will bind, but transcription starts right to the right of this. And the term we use for that is downstream. We say that this region here is downstream of this region here, and inversely, this region here is upstream of this region here, right? Because if you think of it, because what we're going to be doing is transcribing in this direction. So this is the way the river is flowing, and everything is downstream. And so transcription, the gene actually starts being transcribed right here. But this is where the enzymes bind.
The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme.
Okay, so I'm going to back that up for a second so you can write that down. So there are two proteins which are involved in making the enzyme complex that's going to synthesize the RNA. We call it the RNA polymerase core enzyme. The core enzyme is what actually creates the bonds between the nucleotides so that you actually form the nucleic acid. And the sigma factor is needed because it recognizes the nucleotide sequence in this promoter region, so it knows where to bind. So if you didn't have the sigma factor, this guy would not know where to bind on the DNA. So you need both of these together in order to successfully begin transcription. And what that implies is that at the beginning of every gene, it has the exact same sequence. So the sigma factor is able to bind to this specific sequence. It'll vary, but there are similarities and there are differences, but there are enough similarities that sigma factor is always able to find it. We call it together a holoenzyme. So holo meaning whole. So now both together form what we need to make the whole thing work.
DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA.
And notice it's binding to the promoter region.
Complex between RNA polymerase and the DNA molecule.
And notice that we still have a closed double helix. And we're not going to be able to transcribe until that opens up, but at the moment it's closed.
By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript.
Okay, so what's going to happen now is, right, you have the nucleotide triphosphates, and there you've now opened this up. And there's a helicase activity that's part of these guys. So together they have helicase. So it's interesting. It's not like with replication where you have all these separate proteins. We have many different activities located within the same protein. So we have a helicase activity that's able to open this complex up and to start the addition of the complementary bases.
At the 5' end of the transcript, new nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released.
Now, 2 points I want to make here. Number one is we want to remember, how are the nucleotides being added? Are they being added 3' to 5' or 5' to 3'? So let me just bring this up for a second. So if we've opened up the DNA here, right, and here, and we're going to start transcribing in this direction, right? So we're going to start transcribing on this strand. We're going to call this strand the template strand. Oh, I can't do it. Sorry, I have to use the text thing. We're going to call this the template strand. Okay, and we're going to call. Can I do this? Yeah. Nope. Not do that. Sorry. Okay, that's the template strand, right? And then the bottom is called the coding strand. So the bottom over here is called the coding strand. Okay, right here. Okay, so we're going to be building on the template strand. The template strand is the strand you're going to be adding the nucleotides. The coding strand is going to be doing nothing. So if we're building in this direction, and we're going to build a new, no, sorry. Draw, boom, no, draw, boom, and boom, and boom. If we're going to add a second strand, right? So the question is, 3 prime, 5 prime, 5 prime, 3 prime. Which is it? If we're going in that direction, what end? What do we know? What end of this new strand is it? Is this the 3 prime strand or the 5 prime strand? The 3 prime end or the 5 prime? Here, in the new strand. Okay, 5 prime, 5 prime, 5 prime. Okay, no. Sorry, this is the 3 prime end, and this is the 5 prime end. And why is that? Well, what do we know? We always build nucleic acids on the 3 prime end. Remember, nucleic acids, whether it's DNA or RNA, are always built 5 prime to 3 prime. So if we're building this direction, we're building 5 prime to 3 prime. Therefore, what end on the template strand, what end is this? Here. Three prime end. That's exactly right. So this is the 3 prime end, and then this is the 5 prime end. Okay, so good. So we're adding onto the 3 prime end, and then this new guy, this red guy, is adding onto the 3 prime end. Questions on that before I keep going? We're going to come back to that idea when we do the worksheet. But okay, here we go.
As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule, and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex.
So notice that the open complex is a very limited region of only a few nucleotides, right? And it quickly comes back together again. So the holoenzyme opens it up, adds the nucleotides, ligates them together, and then moves along. And so you end up with it only is the open complex, which is here, is a very limited part.
Dr. Hornstein?
Hold on, yes.
Isn't that not the holoenzyme anymore because the sigma factor was released?
Oh, thank you. I forgot to say the other part. Thank you very much. It's no longer the holoenzyme. It's just the RNA polymerase. The other thing that happens, thank you, I got so lost in that. The other thing I wanted to say is, if we go back here, right, is once you start transcribing, you lose sigma factor was only there to recognize the promoter, so we know where to start. Once you start going, you know when to go. Thank you, whoever said that. It's Sequoia. Thank you. Okay, here we go.
RNA polymerase rewinds to form double helix.
Orion, sorry.
Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid, complementary to the template strand.
I just want to make a point here. We have what's called the RNA-DNA hybrid, which is kind of cool, right? For that limited time, while the RNA nucleotides are now joined together and still bound to the complementary nucleotides on the template strand, you have a double-stranded nucleic acid, which is half RNA and half DNA, right? So we call that a hybrid region, right? And you have single-stranded DNA over here because the coding region, the coding strand is doing nothing. That make sense?
Corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule.
Now we're going to get to termination.
Mechanism of bacterial transcriptional termination.
So just to remind you, so remember that there were these two green regions. If we saw at the very beginning, it's at the end of the operon. These are the terminator regions, and there's two of them, and we're going to see why. We've already, where we are now is that the RNA polymerase has already gone through the first terminator sequence, and it's going to hit the second one. And let's see what happens once it hits the second one.
Involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase.
So let me back that up again. This is called the Rho protein. And And the Rho protein is what's going to be responsible for termination. And we want to make a note that this is a helicase. This is a helicase, which means what? Which means that it breaks the bond, the hydrogen bonds between any double-stranded nucleic acid. Okay, so now let's see what happens here. So it slides up, it goes, slides up onto the RNA, it goes around.
Between the row recognition site and the terminator site, a stem loop structure is formed by the sequence in this region.
Now, what happens is, once you get beyond this point here, you get what's called a stem loop structure. And we saw that before, right? And the reason you're able to form a stable stem loop structure is what kind of, type in, what kind of bonds are here between the nucleotides in the RNA molecule? What type of bonds are these?
Wait, Dr. Hornsney, can you rewind the thing a bit?
Yeah, sure.
Okay, thank you.
Sure. So, yes, guys. Okay, so I'll do it. So the Rho protein is going to go over that. And yes, guys, it's hydrogen bonds, right? So we form these temporary hydrogen bonds in the RNA molecule, and that allows for it to form a stem loop structure. And we're going to see why that's important now. So let's just move forward. Let's go ahead and do it.
To the RNA. It is a
So here we go again. It's going to slide over it, right?
Toward the RNA polymerase. Between the row recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA, and the Rho protein then moves to the RNA polymerase complex.
Okay, so the first thing is the interaction between the stem loop and the polymerase. The stem loop then halts the movement. So the RNA polymerase stops moving. And then that allows the Rho protein to slide down here and then do its helicase thing. So, right, so here we go.
Causing the complex to dissociate and terminating transcription.
Right, so helicase is going to, that hybrid that's left at the very end, that hybrid sequence between RNA and DNA, the helicase is going to come and just melt those hydrogen bonds, break it, and then everything's going to go on its merry way. Here's the RNA transcript. It's going to go one place. The RNA polymerase is going to go one place, and then the Rho protein will go somewhere else.
Dr. Hornstein,
Yes.
can you move back just a little? I didn't get that last.
How about if I start here again, and I'll just let it play
That
out.
works out, I guess.
I'll let it play out.
That moves in the 5' to 3' direction toward the RNA polymerase. Between the row recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
Right, and then this is going to be used over again. This will be used over again. And where's this going to go? Where's this? Who's this going to find?
The tRNA.
Right. It's going to find the tRNA and the ribosome, right? Mm-hmm.
And is that what's gonna turn into the proteins?
Right. This is going to be used to make protein. This is going to be used to make protein, right? Again, this is bacteria. So are we in the nucleus?
It's
Are we in the nucleus?
Yes.
Anybody? No, we're not in the nucleus, right? Because
Oh.
there is no nucleus.
Wait.
Right, so
Oh, you're talking about bacteria. Oh, I thought you were talking about in general.
Okay, right. Okay, good. Okay, so in bacterial transformation, what is one major way a determination occurs? Well, we just went through that, and again, this isn't the only way, but it's a nice illustration of how it works. Okay, so that's transcription. Now let's move on to processing, gene processing. So once the transcript is made, different things happen. Turn the page. Different things happen with prokaryotes versus eukaryotes. And what we're going to look at is we're going to compare what is the fate of the RNA after it's made in the prokaryote versus the eukaryote. So here we go.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete.
So what's going on there? This is so cool, right? So they left this part out of the last video because they didn't want to put too much in the same place, right? But what is happening simultaneously in the prokaryote? So let's look. What is this enzyme doing? What is the name of this enzyme? Based on the last, that's right, thank you. It's both translation and transcription are happening simultaneously, right? So we have here the RNA polymerase, and they're not showing what's the name of the protein they're not showing the sigma protein here. They're just showing the RNA polymerase here. And then these are, so what are these little guys here? What are these, the purple guy over here? What is that? What are the two purple guys here? Excellent. These are the ribosomes. And what is the little squiggly blue thing and the little squiggly purple thing? These are the proteins. So simultaneously, before this protein is even done being, before this RNA is being transcribed, you're already starting to make proteins.
Dr. Hunson,
Yes.
is the new RNA edited in a prokaryote or no?
No, that's one of the major differences between them. There's a million of them, but there is no introns and extrons. There are no introns and there are no Try it one more time. There are no introns and there are no exons in prokaryotes. Everything, there is no editing in prokaryotes. Good question. Okay, so let's keep going.
Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins.
I'm going to make this, the next point they make here is that what's one of the major differences between another, and write this down, a major difference is between a prokaryote and a eukaryote, is that the prokaryote, all the genes that are needed for a certain process to take place are on one big RNA, right? So let's look at yesterday's example with betel and tatum. Remember that final thing? We had two different enzymes in that pathway to make asparagine. Was it asparagine? Whatever that final enzyme was. You had enzyme 1 that made, and then you had enzyme 2. And you have that two-step process to go from reactant to intermediate to product. If this was a bacterium, then what would be is that both of those two different enzymes would be on the same transcript here. This would be enzyme 1, and this would be enzyme 2. And so what it does is, so whenever you need asparagine, the bacteria goes in and it makes both enzymes, so it always has both available, so it goes all the way from the beginning to the end. Okay? And this is not true for eukaryotes. Every gene is made individually in a eukaryote. What is an intron and an exon? I'm sorry, I'm jumping ahead a little bit. I thought maybe they covered it in the, did they not cover that in the video, in the Bozeman video? I'm sorry. So all it is that introns and exons, there are parts that are removed from the, introns are the part that are edited out. And exons are the part that are kept there, that are not removed. Okay, here we go.
These clustered genes are referred to as an operon.
So all of the genes that are part of that are for the same function, that are made on that single piece of long RNA, those are called, that's what an operon is. All the genes that are made from one bout of transcription. Okay, now. Now we're looking at a eukaryote, right? And inside the nucleus, right? So we can't do separately. So now we have. So we had transcription here, right? We just saw transcription within the eukaryote, but we have these regions. We have green, orange, green, orange, green, orange, green, orange. And I don't remember the video. Let me just jump ahead, which is which. Okay, so the orange are what are called the introns, and the green are the exons. The orange are going to be removed and thrown away, and the exons are going to be kept there. So let's watch.
I removed and the remaining exons spliced together.
There you go.
A 5' cap and a 3' poly A tail are added.
So in addition to removing the introns, we also add a special cap onto the 5' end of the mRNA, and we add a 3' cap, a tail, onto the 3' end. So we call this the poly A tail, and we call this the 5' cap. And we'll learn when we do the worksheet later what the function of these are, but these serve very important functions. But now, this guy is already made. He's been spliced, he's been capped, and he's added a tail. He's ready now to leave the nucleus.
The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
So there you go. So what happened there was, so notice again, it's only a single protein. As opposed to prokaryotes, you can make several proteins at the same time. And notice in the ribosome clamped here, it made it, and then it left, and the protein is free to go. And notice our ribosome is made of two different pieces, and we'll see that later. And this is the large subunit, and this is the small subunit. Okay, I think that's it for those for that. Okay, wait, Dr. Harnstone. Are the other two PDFs you sent the worksheets? Yes. So before we get onto those worksheets, and either today or starting tomorrow, we're going to do breakout rooms, and you guys are going to do group work. We're going to see how that goes. But before we get there, if you have any questions, I want to finish. This, and
I have a quick question, Dr. Bernstein.
bring that up.
Dr. Bernstein.
Time. Go.
Dr. Bernstein.
Give me 1 second, and then I'll listen. Okay. Go ahead. Question.
What region of the eukaryotic cell does splicing take place and editing? Isn't there a specific region in the nucleus, or
Within the nucleus?
yeah, does it take place or within the nucleus, or?
It takes place in the nucleus. I don't know if there's a subregion within the nucleus. I don't know the answer to that question.
Okay, thank you.
But it does take place in the nucleus, right, as we saw.
Dr. Hornstein?
Yes.
I didn't catch the part where it was talking about the cap and the tail of the eukaryotic primary transcript.
So we're gonna return to this pre-tom in our worksheet in more detail. But there's a special cap that's added to the mRNA after it's spliced, and there's a special tail that's added, and both of those serve an important function, which we'll talk about tomorrow. Okay.
All right, thank you.
Welcome. Okay, so we're looking at this. So what are we looking at here? This is a gene. These are RNA molecules. So my question to you is, what is this a picture of? What is happening here? So first, just tell me, what process are we looking at here? What process are we looking at here? What's the name of the process? Okay, so what I'm seeing is, I'm seeing translation. I'm seeing translation. So if this is DNA and this is RNA, what are we looking at? What are we looking at? It has to be transcription, Orion. Thank you. Right? We're seeing the production of RNA from DNA. Okay? So each one of these represents an independent RNA that's being made from the DNA. So what we're seeing is the RNA polymerase is moving along here, and these guys are coming out and being made. So my next question was at the top, in which direction is transcription occurring? Is transcription going from left to right? Is the RNA polymerase moving this way and churning out all these transcripts? Or is it moving from right to left? Is the RNA polymerase moving that way?
Dr.
So go ahead and write down which is it right, just right to left or left to right. Question?
We can't see your mouse pointing to everything.
What can't you see?
Your mouse pointing to the stuff
You're
on
still
the screen.
sharing the video.
Yeah.
Oh, really? You can't see that?
No.
Sorry.
No.
I thought. Okay, thank you for
You're
letting me
also
know.
cutting out.
Say it again.
You're also cutting out.
Oh, I'm cutting out. Okay.
Oh wait, no, that might be somebody else.
Okay.
I think
Here we go.
somebody.
So let me then, I have to, oh, hold on. That's not what I meant to do. Give me a second. Okay. So annotate is what I meant to do. So we're moving left to right or right to left, and everyone is saying left to right, and that's absolutely true. We are, oh, that's not a good color. Let's try, what's a good color? Dark blue? Oh, that's not. We're moving in that direction, right? And why are we moving in that direction? How do we know we're moving left to right? How do we know we're moving left to right? We're moving left to right because the transcripts are really small here, and they're really big here. So clearly, as the RNA polymerase is moving along here, the transcripts are getting longer and longer and longer and longer, right? And until here, they're just going to drop off. Okay. The last point I wanted to make about this is there and is this. We have something called alternative splicing. And I want you to look at this gene. Can you see the arrow now, by the way? No? Could you just type in?
Yes, I can.
Good. Okay. So here is our gene. Here is our mRNA. What do we mean by the term alternative splicing? You take a look at this and you tell me, what do we mean by the term alternative splicing? Okay, that's a hard one. Another way of splicing. Let's take a look at it. First, we take this gene and we make mRNA from it, right? Bum, 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 bum. Now we're going to splice it. So this is our 1, 2, 3, 4 exons. The rest are exons. So we're going to throw them away, and we're going to throw them away and just connect the exon. However, what's going on here? This mRNA has been spliced, but it's missing one of the exons. It's missing number 4. This guy has also been spliced, but what is he missing? He's missing 3. So this guy is exon 1, 2, 3. This guy is exon 1, 2, 4. So they are alternatively spliced. They have different exons. So the same gene is able to make multiple transcripts. So what is that going to do? Are these the same protein? Is protein A equivalent to protein B? Absolutely not. No, it has different amino acids. When you make this, you're going to be missing any of the amino acids that are coded for by exon 3 are missing here. And any of the amino acids that are coded for by exon 4 are going to be missing in this protein. So these are 2 different proteins. What is the purpose? Well, you're creating multiple proteins in the same RNA. So when we say that there are only 25,000 genes in the human genome, there are actually a lot more proteins than there are genes because you're able to make many proteins from the same gene. So we actually have anybody want to ask a question about that? Someone's cutting out. Oh, Orion, you cut out. Someone's having a brain aneurysm. Okay, could be. Okay. So if chromosomes break and reattach, okay. The other reason that they believe alternative splicing is there is just because if chromosomes, one way to get new combinations of new chromosomes is by recombining chromosomes. You know what? This is a complicated idea. We're going to let it go. But we're going to let that go because I always have trouble explaining that one, and it's not worth it. So, but it's an interesting idea, but it's not been proven yet. Okay. Anyway. Okay. So that's that. So what time is it? Let me see. Okay, it's 11:42. Okay. So let's talk for a second. Let's go back. Let me ask you a few questions before we go. Give me 2 differences between transcription of prokaryotes and eukaryotes. Give me 2 differences between transcription of prokaryotes and eukaryotes. Just give me one. Just give me one. Just type it in. Type in, I'm going to give you 30 seconds to type in one difference between transcription in prokaryotes and eukaryotes. Okay, good. So I saw a lot of good answers there, right? We saw that one, in eukaryotes, transcription happens in the nucleus, whereas it doesn't happen in the nucleus in prokaryotes because there is no nucleus. Because of that, in prokaryotes, again, transcription translation can happen simultaneously, right? And the other thing is the transcript in RNA is altered. The RNA in eukaryotes is altered in three major ways. In a eukaryote, not in prokaryote. What are those three ways that the that eukaryotic transcripts are altered? One of the three ways that you alter the eukaryotic transcript before it leaves the nucleus. Guys, please mute yourself. I'll just mute everybody. Splicing is one. Give me another. Give me two. Give me another one. So there's splicing. What else? Capping is one, and the last one. Tail. The putting on the tail, right? Yes. Good. What else is important? What do we call the region where RNA polymerase binds? Where RNA polymerase and a sigma bind in order to initiate transcription. What do we call that region? Good. And what do we call the area that promotes termination of transcription? What do we call the area that promotes the terminator region? Right. Why do we need two sequences in the terminator region? Why do we need two sequences in the terminator region? That's a hard one. So to allow it to form the stem loop structure that will ultimately knock the RNA polymerase off of the DNA to form that stem loop structure. Okay, so what we're going to do is tomorrow, let me stop sharing. And okay, so tomorrow, what we're going to be doing is we're going to be doing that transcription. We're going to have a double tomorrow. And we're going to do the transcription and part of the translation. No, the transcription worksheet, the transcription in detail, and then we'll start on an animation of translation as well. Okay. I'll stick around for a few minutes. If anyone has any questions they want to ask me about today's lecture or anything else, feel free to stick around. This lecture will be available in 1 to 2 hours on the YouTube channel. And obviously you have access to these animations yourself. Okay. So I'll say goodbye to everyone now, unless you want to stick around. I'll stick around for like.
Wait, I have a question.
Yeah.
I have a question. Wait, how are we supposed to like know the answer to like the attendance questions?
Oh, what was the question? I forgot what I asked. What did I ask you? Good
you
question.
asked about like the structure of the tRNA or something.
Say it one more time.
You asked us what allows tRNA to fold up into a stable 3-dimensional structure.
I showed you that awesome picture yesterday. You don't remember? I'll show you again.
The T thing?
Yes, the T thing. That's right. This. So what I was asking was, what is it that allows this, rather than so tRNA is a single RNA molecule, and yet it folds up into this very complex structure. Why is it? What is allowing that to happen? Or another way of saying that is, what kind of bonds are being formed here?
Okay, there's I don't know, hydrogen peptide that's so far.
Yes.
I'm just naming bonds.
Hydrogen bonds, right? Yes, right.
Hydrogen, okay.
So that's the hydrogen bonds. So that's what allows it to form. So you have these complementary base pairs here and here. Complementary base pairs here and here. Complementary base pairs.
Wait, you're not sharing your screen.
Mr. Hornstein, you're not.
Oh, I'm not sharing the screen. I'm so sorry. Oh, what I
That was
did-
okay.
oh my goodness. Hold on. Here, let me just show you again. Okay. I don't have to make it. There it is. So here. So look, it forms a structure because you have the complementary bases here, you have them here,
Yeah.
you have them here, you have them here, and that's what allows it to form those structures. That's what I was trying to get at with that. Does that make sense, Ella? Ella's already left. Okay.
I have a
Any other questions? Sure. Go ahead.
question. What's the, you know how you said that they're opposite from each other and they have hydrogen bonds and everything, like the CG and everything.
Mhm.
So what about the circular ends, like the like yeah.
Are you looking at this molecule here?
Yeah,
Well,
like the circular part.
yeah. So no, if you, I mean, you look, you'll see there are. Let's take a look here. There are no complementary things going on here. They're not
Yeah, so
each other to do that, right?
what's their purpose?
What is the actual function there? I'm gonna guess. I don't know. We know that this is really important. So I don't know about these two, but the reason it has to be a stem loop structure here is this is the anticodon. This is what's going to bind to the mRNA. And it has to be able to find GCC. So it has to be able to expose the CGG right here in order to form the bonds. And you won't be able to do that unless it's exposed. So this is a really nice way of exposing that. Does that make sense?
Yeah, and also what's the purpose? You know how the tRNA is just supposed to get the mRNA to the ribosome structure and insert it?
Get the amino acid to the ribosome structure.
Yeah, the amino. So like, what's the purpose of the rest of the like nucleotides in the tRNA? Like,
Right. So you want to think of it as having two important ends. This end and this end. This end is the end where the amino acid is attached. And this is the name of the protein that actually attaches it. Okay? So this is where the amino acid attaches. And this is the end where it binds the mRNA. So it's sort of a polar thing. Amino acid at one end, RNA binding site on the other end.
all right, thank you.
You're welcome.
So I have two questions.
Sure.
When are we doing like the worksheets that you posted like today? Like, and do I have to do that for like homework today?
No, you're doing the chapter today.
The chapter outline?
Okay, so do you have the handout that we've been, okay, good. I'm glad you asked me this because it's obvious that it wasn't clear. Let me just move this so I can, do you have, hold on one second.
Oh, well, yeah, I have the thing that we've been working on in class.
Yes, so go to the last 2 pages.
Okay. Yeah.
So what does it say? It says, use the chapter to answer all the fill-in-the-blank questions and all the review questions in the space below. You may make an outline if you prefer. So what I'm asking you to do is, either here or on separate paper, whatever you like, is this is where you're going to go through the chapter. So what you should be doing today and tomorrow for homework is to be answering those questions.
Yeah, I know. But like, when are the worksheets due? Like, do we have
Everything is due. The entire packet is due on Monday morning.
Okay.
Dr. Hornstein?
Yes.
And also, wait, tomorrow, what time is the
Tomorrow's going to be right. Yeah, it's going to be. We're going back and forth now. So tomorrow's going to be 9 and 10.
Thank you.
Cool.
Dr. Hornstein, I have 2 questions.
Oh, sure.
First, for the chapter outline, Is it okay if I do it on Google Docs and submit it as a separate PDF, or does it?
I'd rather you could you just scan the entire thing into one PDF with the outline and everything else?
I don't know how to add. I guess I could try adding it, but I don't know.
Wait, what don't you know how to do?
I don't know how to merge the two together.
Just print it and scan it all together.
I'm really low on printer ink, and there's no coming.
Okay, wait. So you're gonna do a separate outline for chapter 12, and then you have all these other worksheets.
Yes.
How do you normally scan, Sequoia? I'm confused.
Well, what I can do is I can download the Google Docs to my computer, and that makes it a PDF, so I can upload it, or I just scan a paper as a PDF.
Wait, are you printing? I'm sorry, and forgive me if you told me this and I've forgotten. Are you printing out the worksheet and writing on it or not? You're
Yes,
not.
I am.
You are. Okay. So at the end of this, if you're writing on that, can you just, you know how to make a multi-page scanning doc. You can scan multi-pages, right? You know how to do that because you've done that before, right?
Mhm. I don't know how to do it where I have, let's say I have a Google Doc and then I have a scan. How do I merge the two?
So you can't just print out the Google Doc and then scan the whole thing as a single document?
I'm afraid I'm going to run out of ink, and that's.
Okay, it's not the end of the world. You can
Oh
submit.
wait. I have this thing.
I don't want to. It's nice for me to. It's not a big deal if you send me 2 documents. Don't worry.
Okay.
Not a big deal.
Thank you.
You're welcome. Anyone else have any other questions?
I have a
Dr.
question.
Hornstein.
No.
When are the
Yes.
other 2 worksheets due?
Is Tenzin, is you're the one who wants to have a question? Tenzin, do you have a question? Can I see you, Tenzin? Is it possible? I'd really want to see you if I can.
I don't. I didn't get sent the attendance question.
You didn't understand it, or you didn't?
Like you didn't send it to me. I didn't get any.
Hold on.
Like I can't find it in Jupyter.
Hold on. I might have. You're in 5th period class, right? It says you're. Yeah, it's 5th period class. It says that the assignment is April 2nd attendance. Just look at that. April 2nd attendance.
Oh, okay.
Okay?
Oh, yeah, I found it now.
Okay.
Wait, Dr. Hornstein.
Yes, who is that? Joshua?
Josh. Yeah.
Can I see you, Joshua? You guys don't like to be seen. Is it? I really would prefer to be able to see you.
It's not working today.
Okay, go ahead.
The other 2 worksheets due Monday, along with the packet we're doing right now.
I'll make that decision. We're gonna get as far as we can tomorrow.
Okay.
We might have to finish the translation worksheet on Monday, but we'll see how far we get. But you should be working right now. I would be working right now on the chapter right now.
Yeah.
So get that done as much as you can today and tomorrow, so that if I need you to do work on the translation and detail worksheet, you can do that over the weekend, okay?
Okay, thank you.
Sure. Other questions?
I have a question. So for
I can't see you either, Priyanshu? No one wants to show their face? Thank you.
like the introns and exons, is that only in splicing?
What do you mean, is that only in splicing?
Like when you're doing like tapping, or is that also happening? Like I don't really understand the concept of like exons.
Sure. So let me so the concept of splicing is that the RNA ends up being much smaller than the DNA. You make the final RNA. You transcribe the RNA, and then large pieces of it are cut out, and the remaining sections are taped together or pasted together. So we're cutting and pasting some of the pieces of DNA of RNA and throwing away the others. So you're left with a much smaller transcript than you initially started with. And so let me, so yeah, so again, and the example of that would be here, right? So here's our initial transcript, right? It's this big. And now here, I'm sorry, this is the gene. Here's the RNA. But anywhere here, 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 and there, they're cut out and discarded. And all you're left with is 1, 2, 3, and 4, which is, you can see, this is much smaller than that. So that's it. And actually, and this is a little distorted in that the part that's thrown away is usually much bigger than the part that's kept. It's a very small percentage that actually forms the final mRNA.
Wait, but doesn't that like contradict one gene, one protein? Because you're using one gene to make two proteins?
Absolutely right. That's a very good point. So this was discovered after 1952, and we now know that that's an excellent point. Maybe I'll make that the test for the attendance question for tomorrow. It no longer is one gene, one protein for that reason, and there are other reasons as well. But that's one reason. Yeah, that's really good.
Also, do you have like introns and exons when you're like capping, or is that only during splicing?
Do you have them appear web? I didn't hear the first part of your question.
What's the other method of like transcription and translation?
What do you mean the other method?
Isn't there splicing and something else? I forgot.
Is there splicing with something else? I don't know what you're referring to. I talked about capping. I talked about the addition of poly A tails. You were not talking about that, right?
Yeah, when you add tails, like, does that also have like exon and introns?
Okay, let me bring that up. Oh, we don't have a picture of it here. Do I have a picture of it later? I'm trying to think. Give me a second. Oh, so we're going to do this tomorrow, but you can look at it. Here is the RNA. Here is its poly A tail. Here is its 5' cap, and this is the RNA itself. And then this is the tail. A poly A tail does not splice. The poly A tail is just literally, it's a bunch of adenines one after another, and they're all of different lengths depending on the transcript. But it's just a bunch of A's. And the methyl cap, again, is just one single nucleotide up the front, and it's methylated, and that's that. Yeah, so splicing is not involved in either of those in any way.
Okay.
Does that answer your question?
Yes.
Okay, any other questions, guys? Christina, no question? Okay, then this will be, again, in a couple hours or an hour or two, this will be up on the site, okay? Okay, take care, everybody.